I'm Dr. Erika Hamilton, and I lead the breast cancer research program at Sarah Cannon Research Institute in Nashville, Tennessee. And today we're going to be discussing PROTAC, or proteolysis targeting chimera. And specifically for today, this is going to be around the estrogen receptor and breast cancer, although I will mention at the end, this really has quite broad applications. So first, we're going to talk about mechanisms of endocrine resistance in hormonally driven breast cancer. This has been a massive topic with multiple agents in terms of novel endocrine backbones, as well as new combinations and targeted agents, really important in the breast cancer field. We're going to be talking specifically about ESR1 mutations and how we test for these. The PROTAC, the data that was recently presented at ASCO this year on vepdegestrant, or the ER degrader. We're going to talk about this clinical trial data, as well as toxicity profile and tolerability, and then wrap up with a summary as well. So I think we're all familiar with this, but this really tends to continue to be a very big topic in ER-positive breast cancer. We know that endocrine resistance can be through a variety of mechanisms. It can be upregulation of PI3 kinase. It can be RB alterations. It can be loss of the estrogen receptor as well. And then finally, ESR1 mutations. So ESR1 mutations, I like to explain to my patients, is a mutation that tells us that that particular cancer is even more dependent on the estrogen axis. It was so dependent on estrogen that it developed this mutation that's kind of like a light switch now, with the light switch always being turned on and it always signaling through that pathway, even in the absence of estrogen. And so the fact that it can do this in the absence of estrogen really tells you why an aromatase inhibitor doesn't work any longer. And we rely on these novel endocrine agents, degraders, whether that's a SERD or a PROTAC, to be active in these scenarios. We briefly touched on this. I know it's a very complicated diagram on the left, but we certainly can have mutations or amplifications of other growth factor-driven receptors, HER2, EGFR or HER1, fibroblast growth factor as well. Certainly, MAP kinase can lead to resistance with KRAS or BRAF. And then another class of alterations that we do have targetable drugs for, PI3 kinase, AKT, PTEN, with approved PI3 kinase inhibitors, as well as an approved AKT inhibitor in breast cancer. So when we look at ESR1 mutations in breast cancer, it's actually incredibly rare to see this first line or in the neoadjuvant adjuvant curative setting. Our incidence is less than 5% at that point, but becomes very common in endocrine pre-treated patients. So in that second line metastatic setting, you can see on the table over on the right, we can see ESR1 mutations upwards of 40%. And in fact, we actually think that this is probably becoming higher, not lower, as patients are remaining on aromatase inhibitors longer in combination with CDK4/6 than they previously used to. Our median PFS is now really approaching 2 years in the first-line metastatic setting. And so the longer you're on an aromatase inhibitor, probably the more evolutionary drive of these cancers that are highly dependent on estrogen to develop an ESR1 mutation. We can test this both based on tissue as well as blood-based testing. But again, important to recognize that these are not uncommon mutations. We're not talking about a TRK mutation, for example. So who should be screened for ESR1 mutations? Routine ESR1 mutation testing is recommended for ER-positive metastatic breast cancer, specifically for patients that have already progressed on endocrine therapy. Now, in the United States, most of the time, our first-line patients are going to receive an AI in combination with CDK4/6, but this is really regardless of whether patients have received a CDK4/6 or not. And then it's also recommended that timing should be done at the time of progression when you're picking a new therapy, and repeated if disease progresses again, as we know that ESR1 mutations can emerge over time. I'll give a caveat to this. This was a little bit of an older statement, but now we also have results from the INAVO 120 trial, which was looking at a very unique population of patients in the first line metastatic setting who not only had PI3 alterations, but had relapsed while on or within 12 months of their adjuvant aromatase inhibitor. And we now have an approval for inavolisib as a triplet in that first line setting. So the one caveat I would say is that ESR1 is really actionable for everyone's second line. But if your patient has relapsed while on or within 12 months of adjuvant AI, it could actually be your profiling could be actionable in that first line setting, not for ESR1, but for PI3 alterations. And so those are patients that I go ahead and order a broad-based panel for up front. In terms of testing, we have choices in terms of tissue biopsy or liquid biopsy. Liquid biopsy is actually preferred for ESR1 mutations. And that's because when we biopsy a metastatic lesion, we're sticking a needle in one very small area of that lesion, and it only represents that lesion, not other metastatic lesions throughout the body. So as you can imagine, tumor heterogeneity over time, if ESR1 is something that's going to emerge and we stick a needle in one of the older metastatic spots, we may not see that. But when we do a liquid biopsy and we're checking blood for circulating tumor DNA, we're really getting a snapshot of how the entire body looks and are more likely to find that ESR1 mutation that we're looking for. In terms of therapeutic options that are linked to ESR1 mutations, currently FDA approved and commercially available is elacestrant. This was initially presented multiple years ago at San Antonio Breast by Dr. Bardia, but is approved specifically for those patients that had received a CDK4/6, and specifically is approved for those patients that have an ESR1 mutation. We have other combinations that are supported by clinical trial data, randomized clinical trial, and multiple of these are under consideration at the FDA. Fulvestrant abemaciclib, so a post-monarch strategy. We've already have an approval, fulvestrant abemaciclib. This really is a combination that we can use today. But imlunestrant abemaciclib is currently under review at the FDA in the United States, as well as vepdegestrant from the VERITAC-2 study that we're going to be going through today. So this is going to become a very crowded area, and kind of knowing some of the differences between these trials and also the difference in side effect profile of some of these options that we have is going to be important when helping our patients decide what they may want to receive. So let's dive into a little bit specifically about the PROTAC. It's a newer class of drugs. It's probably not one that most oncologists are familiar with. And we can think about this as a degrader, but it degrades in a unique way. It's not just binding the estrogen receptor itself and kind of flagging for degradation. It's using the E3 ligase. And so you can see in our little cartoon here that vepdegestrant is our little squiggle molecule with the red and blue on either side of it. One side of vepdegestrant binds the estrogen receptor. The other side binds the E3 ligase. And so as you can imagine, this brings the two of them in close proximity and essentially ubiquinates the estrogen receptor. And when we ubiquinate the estrogen receptor, that flags it for intracellular degradation. Our proteasome or our intracellular garbage disposal comes in, it's going to gobble up the estrogen receptor and spit it out. What's unique about vepdegestrant is it has iterative potential, meaning that now this molecule is free again. It's gobbled up, it's been spit out, it's free, and it can go bind another estrogen receptor and work again. So we're really able to target multiple copies of the protein, and we're also able to target scaffolding proteins and regulate some signaling pathways that are not otherwise accessible by conventional activators. So file this into the degradation bucket, but with a little bit of a unique mechanism of action. So how does vepdegestrant, our oral PROTAC inhibitor, differ from our oral SERDs or fulvestrant? So fulvestrant has been around since the 1990s. I think we're all well familiar with this. It is also a degrader, but it is not oral. It's administered intramuscularly in 2 injections every 2 weeks for the first month and then monthly thereafter. Some of our patients can struggle with some soreness from that injection site. It has a relatively limited activity against ESR1 mutations, and we now have multiple trials in the second line setting that have shown us in a post-CDK4/6 landscape that fulvestrant as a monotherapy is only giving us a PFS somewhere between about 1.8 to 2.0 months. So clearly not good enough for our patients. Oral SERDs, there are many in development. We currently have one FDA approved, which is elacestrant, but remember that we've already seen randomized data with camizestrant. We've already seen randomized data with imlunestrant that's under review, and so I think this oral SERD space is going to become more crowded. Oral SERDs, again, are as they sound. It's a pill that you're taking at home every day. It binds and degrades the estrogen receptor. It has better bioavailability than fulvestrant. And really, so far as single agent, we've seen the benefit really restricted to those patients that have ESR1 mutations. It has high combination potential, really doesn't tend to have a lot of side effects that overlap with other targeted agents. So everywhere where you think about kind of using an aromatase inhibitor in combination with a CDK, with a PI3 AKT, with novel drugs like CDK2 or KAT6, we could also substitute oral SERDs in this setting. And then PROTACs. Again, oral, again, taken at home by our patients, but it induces degradation via this ubiquitin proteasome system. Again, the bioavailability is very good with this. It may overcome ESR1 mutations and resistance. Very high combination potential. Again, this is a very clean class of drugs in terms of side effects, and we'll get into some of the specifics with the VERITAC-2 trial. And this is not FDA approved yet, but again, based on VERITAC-2, is under consideration at the FDA. So let's go through some of the clinical trial data. This is from the phase 1. We presented this about 4 years ago. This was called ARV-471 at the time, before we had it named vepdegestrant. And at the time, we knew that it targeted both wild type and estrogen receptor mutant. And so we enrolled all patients with ER-positive breast cancer. And you can see the activity over on the right. Our different color bars here represent different doses of vepdegestrant, and you can see that we had very good activity even from very low, down in the 30 milligram range. But you see we went up all the way to 700 milligrams and ultimately decided on the 200 milligrams as the recommended phase dose. Our clinical benefit rate was 40% among our 47 patients that were evaluable. 3 of them had confirmed partial responses, and this was a very heavily pretreated patient population. You can see everyone had seen CDK4/6. Almost everyone had seen fulvestrant. We had many patients, 2/3 or more, that had seen chemotherapy, and even some of them had seen an investigational SERD, if you look at these little dots on the left of the bars. And even among our patients that had seen an investigational SERD, we saw some patients that had good activity, telling us that this was a class of drugs that could have activity even after the oral SERDs. In the phase 2 cohort expansion of the VERITAC trial, we looked essentially at 2 doses, 200 milligrams once daily or 500 milligrams once daily, and we were a little bit more restrictive on who could come on to this trial. Patients had to have had at least 1 prior endocrine therapy for metastatic breast cancer. They had to have had CDK4/6, and they did not have to have chemotherapy, but were allowed to have up to 1 line of chemotherapy. Now, I'll tell you to be quite cautious when you're comparing, cross-trial comparisons here, because not all of the oral SERD, PROTAC, et cetera, trials had required a CDK4/6 inhibitor. And so this was a uniform population, and I think this is important when we're comparing PFSs across compounds. So in terms of who enrolled here, everyone had seen CDK, 79% had seen fulvestrant, and almost half had seen chemotherapy. And our ESR1 mutation rate of patients that were coming onto this trial ended up at about 58%. We saw a clinical benefit rate very similar between the two doses in this high kind of 35 to 40% clinical benefit rate. And then if we were enriching for patients that had ESR1 mutations, again, there's nothing magic about an ESR1 mutation, but really enriches for patients that are more likely to still have endocrine sensitivity in their tumors. We were able to boost this clinical benefit rate up above 50%, so 51.2%. In terms of progression-free survival, when we looked at all patients, this was about 3.7 months. But again, we were able to optimize this to be a bit higher if we looked at those patients that had ESR1 mutations, again, 5.7 months. So similar to elacestrant in terms of having an ESR1 mutation predicted a greater likelihood of benefit and a longer PFS on vepdegestrant. So clinical benefit rate, again, in the ESR1 mutant subset was higher at both doses that were tested. Fatigue and nausea were actually our most common adverse events, but really no higher grade adverse events with vepdegestrant. Very intriguing, and we were looking forward to seeing this in a larger registrational trial to really get a better idea of the tolerability of this. Ultimately, 200 milligrams once daily was selected as the phase 3 dose, as we really didn't see any differential activity between the 200 and the 500 milligram dose. Just to show you the ability of combining with other agents, this was vepdegestrant in combination with palbociclib, so our CDK4/6 inhibitor. Neutropenia, not surprising, was the most frequent adverse event. This was about 90% grade 3 and grade 4, which was a little bit higher than we would have anticipated seeing. And really, this came twofold. One, these patients were very heavily pretreated. They had already seen CDK4/6, et cetera. And so I think didn't have as robust of a bone marrow reserve. And we also saw a little bit of a drug-to-drug interaction where our exposures to palbociclib was higher for those patients that were receiving vepdegestrant. So we probably didn't need to come in at the 125 milligram dose. But ultimately, we saw clinical benefit rate in essentially 2/3 of patients, over 70% for those patients that had ESR1 mutations, which was really quite compelling. A median duration of response over 10 months in those 13 patients that were responders, and a progression-free survival that was approaching a year at 11 months, regardless of whether patients had an ESR1 mutation or had wild-type disease. So I found this last bit of this tidbit very interesting because before that, with the oral SERDs, we really hadn't seen equal benefit in those wild-type patients. So we had this gestalt that vepdegestrant in combination with CDK potentially could overcome the need to have an ESR1 mutation. Ultimately, all of this data led to the VERITAC-2 study, which was our phase 3 registrational trial of vepdegestrant versus fulvestrant, a very straightforward design. Patients had to have had prior endocrine therapy in combination with a CDK4/6. They could have received up to one additional line of endocrine therapy. They could not have received prior fulvestrant or prior chemotherapy for metastatic disease. And it was a 1-to-1 randomization to vepdegestrant orally once daily or fulvestrant at the standard dosing, 500 milligrams intramuscularly, days 1 and day 15 of cycle 1, and then day 1 of each subsequent cycle. Our primary endpoint was progression-free survival by blinded independent central review initially in patients that had ESR1 mutations. If that was positive, then we would be testing all comers, so regardless of the mutation. And then key secondary endpoints, overall survival, again, was going to be tested ESR1 first, then everyone, as well as things like clinical benefit rate and overall objective response rate. So who were the patients that enrolled on this trial? You can see median age was about 60. We had about 50% of patients were Caucasian. We had a high percentage of patients that were Asian because this did enroll in many countries throughout Asia. And we had lower representation of African American patients. In terms of ESR1 mutations, the overall was 43% of patients entering the trial had an ESR1 mutation. And in terms of sites of disease, about 2/3 of patients had visceral disease, almost half had liver metastases, and it was pretty rare, less than 20% of patients having bone-only disease. If you look over on the right side of the screen, you can see that about 80% of patients had only received one prior line of therapy in the metastatic setting. And we know based on the inclusion-exclusion criteria, this had to be endocrine therapy in combination with a CDK4/6, but about another 20% had received an additional line of endocrine therapy before coming on. You can see, again, universally, everyone had seen a CDK4/6 inhibitor, and you can also see the breakout of prior CDKs, about 50% palbociclib, about 30% ribociclib, and about 20% of abemaciclib. So actually a pretty good representation across the three CDK agents. Again, if we're looking at co-primary endpoints on this top left, you can see progression-free survival 2.1 months with fulvestrant, so very in line with what we've seen from other randomized trials, whether Emerald, et cetera. And then vepdegestrant 5.0 months for patients that had an ESR1 mutation. This was a hazard ratio of 0.57, highly statistically significant with a p-value of less than 0.001. If we look on the bottom, so again, now among all comers, regardless of ESR1 status, we see 3.6 versus 3.7 months. This is not statistically significant. So again, we have another trial where the benefit for the novel endocrine agent is restricted to those patients that have an ESR1 mutation. So when we go on to look at subgroups, I think that there are certainly some areas in terms of geographic region, et cetera, where there's smaller numbers, but we were not able to pick out a population that did not look like they benefited from vepdegestrant, whether patients were older or younger, pre or postmenopausal, whether they had visceral disease or not, and really whether they their prior therapies had included one or two prior lines. Benefit really looked quite consistent. So ESR1 mutation being the factor that predicted benefit to vepdegestrant. In terms of safety and tolerability, I think this data was really quite interesting. You see that vepdegestrant and fulvestrant in terms of grade 3 adverse events or greater really not looking very different, about 20-ish%. And then when we start to dive into treatment discontinuation or reduction, really exceedingly small. 3% of patients discontinuing vepdegestrant, only 2% having a reduction. And so when we group that together, either a dose reduction or a discontinuation of less than 5%, this is certainly pretty compelling. This is telling me that patients are actually tolerating this quite well, and specifically tolerating it better than we see some of our combinations with PI3, AKT, combinations with abemaciclib, for example, where patients have more side effects. To get into what these side effects were, if we look over on the right side of the screen, you can see fatigue was actually the most common. About a quarter of patients experiencing some grade fatigue, about 16% of fulvestrant. But said a different way, 3/4 of our patients with metastatic breast cancer were not even experiencing any grade fatigue, and none of this was severe. You can see that our second most common adverse event was AST/ALT increase, only at 14%. Again, almost universally grade 1 here. And then I also want to just point out GI side effects. Nausea, 13%, essentially all grade 1, 0 grade 3, grade 4. And then the other GI toxicity that you'll not see represented on this table is diarrhea. This was actually less than 10%, so it didn't even show up on the table. So again, a very friendly side effect profile in terms of gastrointestinal side effects, which I think means that it would be a very good combination partner as well. So again, the phase 2 VERITAC study, you can see fatigue in about 23% of patients, AST/ALT increases, hot flashes in only about 20% of our patients. So really a mild toxicity profile in summary, very few grade 3, grade 4 adverse events. And then I also wanted to address QTC, as this can be an issue with some of the endocrine backbones. We initially thought in the QTC sub-study of almost about 90 patients that there was a very mild increase of about 11 milliseconds in the QTC F from baseline. This was dedicated and studied and ultimately came back with there is no large QT prolonging effect. This was less than 20 milliseconds, even at the higher end of the upper limit of the 95% confidence interval. And so there are no mandatory EKG monitoring or any of this that we need to worry about patients that may be receiving vepdegestrant in the future. So in summary, PROTACs are active in patients even that have pretreated with oral SERDs. Vepdegestrant is our first PROTAC inhibitor in breast cancer to go through a randomized phase 3 registrational trial and showed a clinically meaningful, significant, statistically as well, improvement in progression-free survival versus fulvestrant. So 2 single agent endocrine therapies among those patients that had ESR1 mutations. Our overall survival data is still immature, follow-up is ongoing, and vepdegestrant really standing out to me in terms of very few dose reductions or discontinuations, telling us that patients tolerated this quite well. And our pressing need still in ER-positive breast cancer is figuring out who post CDK4/6 still has endocrine sensitivity and are very appropriate for these further endocrine strategies versus who may have endocrine resistant disease and should actually move on to drugs like antibody drug conjugates, for example. There are multiple other clinical trials with vepdegestrant. You can see some other combinations that have completed accrual, a combination with everolimus, a combination with a CDK4 inhibitor. There was also an umbrella trial called TACTIVE-U that looked at ribociclib, abemaciclib, kind of our ability to combine with CDK4/6 inhibitors. And VERITAC-2, our number one here, is currently at the FDA for consideration of approval based on VERITAC-2 data. So I'll also give you maybe just a bit of a teaser of where else you may be seeing PROTACs moving forward. Certainly we have ER-targeted agents. There's a couple other compounds in development. Androgen receptor is another big one. And so some of our androgen receptor degraders, particularly being looked at for prostate cancer, as well as some of the liquid side of oncology, B-cell malignancies, as well as lymphomas, looking at BTK as well as BRD4 degraders. And then finally, to leave you with one other exciting a diagram is some of the other places that PROTACs actually can be used. So I'm going to familiarize you with a couple of terms. These are not ready for prime time yet, but I think really just shows where we can go with this technology. ClipTac is essentially a 2 precursor molecule that can click intracellularly, and it's smaller and then can kind of make one large PROTAC once it's within the cell of interest. Photoprotacs, so trifunctional molecules that have not only the ligand for E3 ligase, a photo switch, and a ligand for the protein of interest. So you can imagine this can be inactive in the dark, then can become active under blue-violet light. So you can almost think about it as maybe like a radio sensitizer, but something that you can turn on and off in the presence of light. That would be very interesting, and we could therefore kind of affect which tissues it could be active in. And then also an antibody PROTAC conjugate. So delivering a PROTAC to antigen-expressing cells, whether that's trastuzumab, linking HER2, et cetera, with the E3 ligase and being able to degrade specifically in cells that have this. So very exciting, and I think a lot of new options on the horizon that will be in development.
Subsequent to the recording of this program, the FDA granted approval to imlunestrant for patients with ER-positive, HER2-negative, ESR1 mutated, advanced or metastatic breast cancer with disease progression after at least one line of endocrine therapy. The approval was granted on September 25, 2025, based on data from the EMBER-3 trial.